Well, let's attempt the integumentary system. You probably are well aware that the integumentary system features the skin, but also included are hair, nails, and the many sort of accessory structures that we find in the skin, including glands, but not limited to glands. We already have established that the skin features two layers: the epidermis, which is outermost or most superficial, and the dermis, which is innermost or deeper. Now, the hypodermis is not actually part of the skin. It underlies the skin and is therefore in contact with the dermis. It's not in contact with the epidermis. It's also known as the subcutaneous layer, and the hypodermis is made primarily out of adipose tissue, okay, for insulation, cushioning or shock absorption, and to some degree anchorage, okay. But epidermis and dermis are, as we very recently established, made, what's the word I'm thinking of? Respectively, out of epithelia for the epidermis, specifically keratinized stratified squamous epithelium, and connective tissue for the dermis, specifically loose areolar connective tissue and dense irregular connective tissue. The dermis is vascularized, whereas the epidermis is not, and therefore the dermis is acting to at least some degree as life support for the epidermal cells that are living. I'm on slide 4, and we're going to use this image over and over and over again because it actually provides a lot of detail for us. Here, underlying the skin is that adipose region, that hypodermis, that subcutaneous layer, okay? Here, this pinkie pie region is dermis. Here is epidermis, and the illustrator, at least at this scale, has made a fairly subtle attempt to show a difference in texture between dead keratinized stratified squamous epithelium and living keratinized stratified squamous epithelium. Okay, but if we zoom in, that becomes much more evident. So outermost, wow, that's not even legible, Tessa, come on. Let's try again. Outermost is dead. Innermost is living. So the living cells with the epidermis are in contact, at least some of them, with the dermis. Now you probably noticed that the illustrator made an effort to make some of the structures that are housed in the dermis appear as if they are made out of the same stuff as epidermis, and that's a very purposeful decision on the illustrator's part. These structures, here's a sweat gland, or really the secretory unit of a sweat gland, whereas this is the duct, but together, sweat gland. Okay, here's a hair follicle. All right, and these sort of nubs associated with the hair follicle are sebaceous glands. Okay, Okay, sweat glands, hair follicles, sebaceous glands, they are all derived from epidermal tissue, even though they're housed in the dermis. We'll come back to that later, but students often struggle with that, so I wanted to bring it up sooner than later. I'm moving on to slide 5, and we're going to start really focusing on the epidermis, and then once we're done with that focus, we'll further delve into the dermis. There are certain cells that we can predictably find in the epidermis. Most of the cells of the epidermis are keratinocytes. Okay, and keratinocytes are called keratinocytes because their specialty is the production of keratin. Keratin is a protein, a somewhat fibrous protein that is strong, but is also waterproof. It's waterproof, and it helps to therefore prevent desiccation. However, keratin alone isn't enough to ensure that the body doesn't lose water through the epidermis. Between keratinocytes, at least those that are apical most or surface most, in other words, those that are dead, we can find sort of like a grout, if you will, that's comprised of glycolipids. And glycolipids, that grout, ensures that water can't seep or leak in between keratinocytes, right? Whereas keratin in keratinocytes, even those that are dead, ensures that water isn't lost through the cells, okay? So between keratin and glycolipids, we have a waterproof, not a totally impermeable covering, but a waterproof and somewhat selective covering, okay? The keratinocytes feature an abundance of tight junctions, okay? We lose millions of keratinocytes normally, a healthy body does, every day. And as you can imagine, that's okay because epithelia have regenerative superpowers. Okay. We can also find them on slide 6 in the epidermis, cells that are called melanocytes. And melanocytes, their bodies are sitting in the deepest or innermost single layer of the epidermis. I'll come back to that later. I know that doesn't really make sense right now, but deep in the epidermis. And a melanocyte body looks pretty much like any other animal cell, okay? However, it's got oodles and oodles of projections, and these projections are, for the most part, reaching toward the apical most surface. They don't actually reach that far, but that's the direction that they're reaching. And they're sort of working their way in between keratinocytes, and thereby delivering sort of door-to-door delivery the products of melanocytes. And the product of melanocytes is, not surprisingly, melanin. Melanin is a pigment, and pigments are proteins, so we're hopefully not surprised that keratin is a protein, melanin is a protein, hemoglobin is a protein, antibodies are proteins, hormones are proteins, right? The body specializes in making proteins and lots of different kinds of proteins. Well, the boxes, if you will, the packages that melanin is placed into are called melanosomes, okay? And melanocytes use their almost spider leg-like projections to shunt melanosomes into the spaces where slightly more mature keratinocytes lie, and thereby deliver melanin. When a keratinocyte takes up that melanin, it will shunt the melanin, not just store it in the cytoplasm, but actually shunt it to the nuclear envelope, the nuclear membrane, the membrane around the nucleus, or the membrane that establishes the organelle called the nucleus, and specifically to the sunny side of the nuclear envelope, which is just brilliant, just mind-blowing that these cells are protecting the DNA within only on the side that's exposed potentially to UV, and thereby melanin acts as a sunscreen and a cellular sunscreen for just DNA, because UV damage is a huge mutagen. UV can cause mutations. No, thank you. So really, really cool. We can also find dendritic cells, also known as Langerhans cells, in the epidermis, and these guys are found in the deeper layers of the epidermis, not necessarily deepest, but deeper layers, and they're kind of like the private police force of the epidermis. In other words, they have immune function, okay, but they're housed only in the epidermis, okay? We can also find Merkel cells. It's a Merkel in the epidermis. They are in the deepest. Whoops, I just misspelled deep. Good grief. That takes talent. There we go. The deepest epidermis, okay? And they appear very much like a chair cushion, and that cushion is sitting on top of nerve endings, and that anatomy allows Merkel cells to sort of relay any pressure changes that are caused by touch, okay, to those nerve endings, and therefore Merkel cells are acting as sensory receptors. Okay, moving on to slide 7. This image is very helpful. We're going to label this particular image just all to tomorrow. So right now it looks really, really clean, but we're going to make a mess out of it. So this one, this side, the left side, this is a micrograph. So this is an actual photo, okay, whereas this is an illustration. Okay, and sometimes features are easier to see in an illustration because it's idealized than they are to see in a photo, but we'll kind of bounce back and forth between these two. So here is kind of a little finger of dermis that's reaching up into the epidermis, and that's very common, that we would see kind of a rippling interface between dermis and epidermis, and we'll come back to that later. But a long hugging along that kind of finger is a single layer of cells, okay, that includes, but is certainly not limited to, the cell bodies of melanocytes, and I've just circled a cell body of a melanocyte, and it's really difficult to see in this illustration because the illustrator chose a really pale gray as the color code here, so I'm just really emphasizing these projections, the spider-leggy projections of this melanocyte reaching apically. This is apical surface here. Okay, out among and between keratinocytes. All of these, I don't know, sort of orange, peach cells are keratinocytes, or each is a keratinocyte. Well, that's a ridiculous N. Okay. Here also sort of interrupting or embedded in that innermost layer is a Merkel cell sitting on top of a nerve ending. Okay. And then here, not quite as deep, in other words, not limited to that innermost layer, is a cell that looks kind of like a macrophage, because it is. Okay, it's just a private macrophage of the epidermis. This is a Langerhans cell, or a dendritic cell. You can use either name, but in 241, we're going to use the term dendrite later. In 242, we're going to use the term Langerhans. So, to mean totally different things. So, in order to not confuse yourself in 241, you might want to refer to this as a Langerhans cell for now. Really, there's no way to make this absolutely frustration-free. Okay. Notice that the illustrator made an effort to make this section here look very sort of desiccated. There aren't any nuclei evident, and that's because these are dead, okay? Whereas this portion of the epidermis is living. Okay? That means that these keratinocytes are the living. Keratinocytes are busy making keratin, whereas these keratinocytes, those that are dead, can no longer make keratin. They're they're dead. How can they produce if they're not alive to make proteins? Okay, we'll come back to that. Now I'm going to move along to slide 8. There are up to 5 layers or strata. A single layer would be a stratum, okay, that make up the epidermis. And the reason why I say up to 5 is that all of our skin is guaranteed to have 4 strata, whereas only the skin on our palms and soles will have a 5th layer, a 5th stratum, okay? So the guaranteed layers are stratum basal, stratum spinosum, stratum granulosum, and stratum corneum. The sometime layer is stratum lucidum. All right, so any skin, no matter what, will have stratum basal, stratum spinosum, stratum granulosum, stratum corneum, actually in that order from deep to superficial. Okay, deep to superficial. Whereas palms and soles will have stratum basal, stratum spinosum, stratum granulosum, stratum lucidum, stratum corneum in that order from deep to superficial. Okay? These 5 strata are at least slightly different anatomically and therefore slightly different functionally. All right? We tend to refer to the skin on the palms and the soles as thick skin only because it has those 5 layers. But that extra thickness is actually somewhat laughable, since stratum lucidum is, on average, probably about 4 layers of cells thick. So it's not that much more impressive, okay? Whereas any other skin covering the body would be referred to as thin skin, which doesn't mean more vulnerable necessarily, okay? Moving on, I'm on slide number 9, and this is that same figure, but here we've labeled the different strata that would be present in all skin. In other words, lucidum is not depicted, okay? The region that we identified as being dead just a moment ago is stratum corneum, okay? And the innermost region that includes, but is not limited to, the bodies of melanocytes and the bodies of Merkel cells is stratum basal, okay? Immediately overlying stratum basal is stratum spinosum, and I'll establish for you why it's named spinosum when we get a little further. And then the layer between spinosum and corneum, which is depicted here as quite thick. It can be actually a lot thinner, relatively thin. It is stratum granulosum, okay? By the time a keratinocyte is mature enough that it's moved into the stratum granulosum, it is just about to die, and it is getting rather full of the ingredients to make keratin, as well as getting rather full of glycolipids. Those glycolipids will be extruded into interstitial space to act as grout in the stratum corneum, okay? If we use this idea of keratinocytes moving from basal surface to apical surface, then it might become more clear to you that the stratum basal is where keratinocytes are, if you will, born, okay? So they're youngest in stratum basal, and certainly oldest where they're shed at the apical surface, okay? And because stratum basal is continuously performing regeneration, we have this continual aging and moving process where keratinocytes are being shunted toward that apical surface. Moving to slide 10, we'll have a private or semi-private slide for the strata. Stratum basal, again, is deepest. It's the deepest layer. It's the only layer that's in contact with the dermis, okay? It will be comprised primarily of keratinocytes, but they're stem-like keratinocytes that are constantly undergoing mitosis, constantly dividing to make daughter cells. One of those daughter cells will stay behind and simply replace the parent, okay, act as a new stratum basal cell, and the other will be pushed toward stratum spinosum, and then stratum granulosum, and then stratum corneum, okay? And it takes about a month for that keratinocyte daughter that is going to proceed through the layers of the epidermis to get all the way from stratum basal to the apical most surface where it's actually shed, okay? Melanocytes, we expect to sort of interrupt stratum basal, or at least their bodies, okay? And we're also going to find intermittent Merkel cell bodies here. Moving to slide 11, here's that same image with stratum basal again labeled, and I've placed all of the pertinent text on that image just to kind of keep everything together. On slide 12, the focus is stratum spinosum. Stratum spinosum is kind of a frustrating layer because the name really refers to what the cells would look like if they were dead, and they are not dead in the human body. So this is the layer that's just superficial to stratum basal. These are sort of like newborn keratinocytes, or at least young keratinocytes, okay? In the living body, if we were to look at those keratinocytes neighboring each other in stratum spinosum, okay, we might be able to see, oh, okay, yeah, there are lots of intercellular junctions in place. And I'm really focusing just on this central keratinocyte, okay? This arrangement, these cells, they look nice and plump, okay? But if I were to take all these cells out of the body and allow them to desiccate, then these intercellular junctions, which are not strictly tight junctions, but certainly tight junctions are abundant in the epidermis, okay? These intercellular junctions, they would still hold these cells together, but the cells would kind of draw, away from each other. I need more colors. I wonder, will this thing give me colors? There's only one way to find out. Oh, yes. Okay. Let me undo that and give you some colors. Uh-oh. Make it a little messy, or less messy. A little messy. Okay. When this guy desiccates, you even see that green. Okay. The now dried out, withered keratinocyte looks kind of spiky. And that's actually where the name of this stratum comes from, is that if these cells were allowed to desiccate, they would be spiky, or they'd have spines, which is, I agree, silly, but that's where the names come from, okay? So the name really just refers to keratinocytes once they're desiccated, which is goofy. We're going to have the extensions of melanocytes reaching out into stratum spinosum, therefore delivering melanosomes, the packages of melanin, okay? Langerhans cells or dendritic cells are going to be hanging out primarily in stratum spinosum, okay? And stratum spinosum is several layers thick, whereas stratum basal is only one cell layer thick, okay? Slide 13, same gist, where we've labeled again, but this time put all the pertinent text with that label. Slide 14 focuses on stratum granulosum. And stratum granulosum is typically thinner than stratum spinosum. It's 1, 2, 3, or 5-ish cell layers thick, all right? And this is where the process of keratinization will really begin and end. Keratinization is the filling of a keratinocyte with keratin, and therefore also the murder of a keratinocyte, because once it's full of keratin, there isn't room for cytoplasm, there isn't room for organelles, so the cell gets overrun by keratin, okay? In the process of sort of aging through the stratum granulosum, these keratinocytes, they're going to flatten out. In other words, they're going to assume their squamous shape, that shape that we see apically, okay? The organelles are going to be displaced, the cytoplasm is going to be displaced, and we're going to accumulate vesicles, okay? Those vesicles will house two things: the ingredients for making keratin and glycolipids. So some vesicles will have the ingredients for keratin, and some other vesicles in the same cells will have glycolipids. They'll contain glycolipids. Okay? Well, as soon as we put the ingredients for keratin together in the same vesicle, we're going to get keratin. Okay? So it's a little bit hair splitting to bother naming those ingredients to make keratin. But as the granulosum cells load up with these vesicles, they end up looking kind of polka dotted. They're so full of vesicles that they look like, I don't know, they've got chickenpox. Okay? And therefore, they kind of look like they're filling up with grains of sand. That's why we often refer to these vesicles as granules. Okay? And now you can see where this stratum gets its name. Okay? Those granules. Some granules with the ingredients to make keratin, some granules with glycolipids. Okay? Once cells reach that border between granulosum and any superficial layers, they are just about dead. On slide 15, same image, again with stratum granulosum specific text. On slide 16, stratum lucidum is the focus, but recall, please, that this is only a sometimes layer. It's that one that we find only on the palms and the soles, the palms of the hands and the soles of the feet. Okay. Very, very thin, 2, 3, maybe 4 layers of flat, dead keratinocytes. By the time they've left their granulosum, they're dead. Okay. And this is, if it's present, this is where the ingredients to make keratin will sort of snap to. They'll come together and keratin will be formed. Okay. When lucidum isn't present, which really over most of the body it isn't, this same combination of ingredients will instead happen in the stratum corneum. Okay, and that's actually the next slide. Slide 17 is stratum corneum. The stratum corneum is really a huge stratum of the epidermis. It's comprised of 20 to 30-ish rows or layers of dead cells, and therefore makes up the bulk of the epidermis. All right. There are 2 reasons why the cells, once they've reached this layer, are guaranteed to be dead. They're dead because they're so full of keratin, but they're also dead because they're so far away from life support. Okay, so it's kind of like they're going to die one way or another. So all dead, but still very functional. When keratinocytes fill with keratin, they don't lose that keratin when they die. They stay more or less intact and full of keratin. So they end up being sort of like flakes that are made out of keratin. And in between those flakes, we seal up those otherwise leaky spaces with glycolipids. And glycolipids are lost or extruded from keratinocytes just before they die. Okay, it's really stratum corneum, therefore, that is responsible for waterproofing. Okay, again, waterproofing via keratin and glycolipids. All right. The cells in the stratum corneum, they even though they're dead, they don't break up, and in part that's because the keratin inside is sort of holding them in their squamous shape. okay? We do eventually lose them, but they flake off, they slough off. We lose about 50,000 keratinocytes off the apicalmost surface of our skin every minute. Most of the dust in your home is comprised of these flaked off dead keratinocytes. Surprise! And the reason why you can't get rid of dust mites is because you keep feeding them. Gross. Or yay. Slide 18, same visual support. Slide 19, here we get to start talking about the dermis. The dermis, as you know, is comprised of connective tissue. It is innervated. It is vascularized. It features lymphatic vessels, whereas the epidermis does not. It houses hair follicles, sebaceous glands, and sweat glands, which, even though they're housed in the dermis, they're derived from epidermis. They're made out of sort of epidermal tissue, if you will, okay? There are two layers to the dermis, rather, and we started previewing them already. The papillary layer and the reticular layer. The papillary layer is relatively thin, and the reticular layer is relatively thick. The papillary layer, we already established, is made out of loose areolar connective tissue. Don't abbreviate on exams. And the reticular layer is made out of dense, irregular connective tissue. Okay. On slide 20, we're seeing that image that we saw early on again, but this time it's hyper-labeled so that we can see a little bit more of how busy the dermis is, how many accessory structures there are housed in the dermis. It's sort of overwhelming. So again, here's our epidermis, okay? Here's our dermis. This guy's just hypodermis, which is actually not part of the skin, okay? And if this is dermis, then just this beep is papillary layer, whereas the rest of it is reticular, all right? We can find sweat glands here. We can find sebaceous glands here. We can find hair follicles here. We can find muscles that are called erector pili muscles. We'll come back to that. We can find certainly blood vessels here, okay? Nerve endings, for instance, this is a nerve ending. This is a nerve ending. Heck, this is a nerve ending. Housed in the dermis, and many sensory receptors housed in the dermis. Oodles and oodles. Okay, and here is that kind of wavy or ripply interface that I mentioned a few slides ago between dermis and epidermis. These little finger-like projections of the dermis are called papillae, and now it's probably easier to understand why that uppermost layer is called the papillary layer. Moving on to slide 21. Slide 21 is really important because the way that this image is both stained and colorized really helps drive home how different the layers of the dermis are in appearance from each other, but it also helps me to explain to students what is and isn't epidermis. So often this layer that's colored this orangish hue, students will identify this as epidermis, and this does belong to epidermis, but this is not the entire epidermis. This is just stratum corneum. Okay, but this region that's colored blue, not literally in your body, but here, colored or colorized blue. This is the living portion of the epidermis. So this is epidermis as well. Lots of students misidentify that as dermis. It's not dermis, it's epidermis. So this includes the other strata, stratum granulosum, stratum spinosum, stratum basal. All right, stratum basal would be this thin single layer here. Okay, in other words, stratum basal is hugging or in direct contact with dermal papillae. Here is a dermal papilla. All right. Now, the dermal papillae are made out of loose areolar connective tissue because they belong to the papillary layer. Papillary layer is relatively thin, and then everything else, in fact, even beyond this slide, would be reticular layer. And you can really see how much more densely packed the fibers are in the reticular layer than they are in the papillary layer, because the papillary layer is comprised of a loose connective tissue and the reticular layer is comprised of a dense connective tissue, all right? Whereas epidermis is, of course, comprised of an epithelium, keratinized, stratified, squamous epithelium. Slide 22. The papillary layer, again, comprised of loose areolar connective tissue, provides lots of space for macrophages and other white blood cells to roam. Okay, so that they can sort of act as sentries. They can act as guards, if you will. In other words, no thank you, please do not enter here. Dermal papillae are shown in this illustration that somebody very talented made. I'm kidding. These are dermal papillae. Okay? And dermal papillae definitely increase the surface area that sort of anchors the dermis and epidermis together, all right? But we also have these much more pronounced, I guess I shouldn't say pronounced, they're actually more subtle, larger kind of ridges or humps, right, in the skin. And these are, unfortunately, labeled differently. So this guy here is a dermal ridge. Dermal ridge. Okay? And this guy here is, actually I'll just borrow the typing here, is an epidermal ridge. Okay? Together, a dermal ridge and an epidermal ridge is called a friction ridge. And it's friction ridges, not dermal papillae, that are expressed as fingerprints. Okay? Now, notice that included in the illustration is a tube and a little sort of ball shape. Okay? Maybe you see automatically a duct and a secretory unit. In other words, these are sweat glands. Okay, they're specific sweat glands, but we're not quite ready to explore that yet. I'm trying to write super tiny with this pencil that does not really allow fine writing. Okay, anywho, notice that the duct of these sweat glands leads to the very apex of that friction ridge, okay? That means that our fingerprints, the ridges that make up our fingerprints, have along their curvatures sweat pores. And that sweat then acts as a sort of ink, so that whenever we touch something, our fingerprints act as the stamp, and our sweat acts as the ink that leaves the pattern of our fingerprints behind. And it might be easier for you to understand that if we move to slide 23, where we're looking at those friction ridges and seeing those very uniformly placed, very centrally located sweat pores, okay? Friction ridges, in other words, fingerprints, are the stamp, and the sweat is the ink that we leave behind on things that we touch. Just really cool. All right, moving to slide 24. The reticular layer makes up about 80% of the dermis. Okay, again, it's the bulk of the dermis. And it is, as you already know, comprised of dense irregular connective tissue. All right. This is where the bulk of blood vessels are found, the bulk of lymphatic vessels are found, the bulk of at least larger sensory receptors are found. We can find adipose cells, meaning adipocytes, here and there through the reticular layer, whereas the hypodermis, which underlies the reticular layer, is made almost entirely of adipocytes, all right? We can also find, obviously, lots of collagen fibers in the reticular layer, considering that it's dense, irregular connective tissue, but, of course, running in multiple directions. Therefore, the reticular layer helps the skin to withstand multi-directional force or multi-directional tension, okay? Wherever these collagen fibers are less densely packed together, they form what are called cleavage lines. And cleavage lines are actually quite predictable. In other words, we know from obviously lots of study of the human body that the sort of less densely packed fiber areas are here and not there. We have mapped all those sort of more sparsely grouped areas. And surgeons use these predictable regions, these cleavage lines, when making decisions about where to cut, because those regions are going to heal more readily, and certainly yield more readily. This is actually a map of cleavage lines on slide 25. Good grief, don't memorize it. It's not like I'm going to show it to you again and say, what is the name of this one cleavage line? Oh my God, no thank you. But very predictable, kind of surprising. Let's go to slide 26. We're still in the topic of the dermis, right? Just in case we haven't already introduced enough lines to you, we need to know what flexure lines are, not to be confused with cleavage lines. Flexure lines are folds and creases that form more or less permanently over joints that we use a lot, okay? And because we use these joints a lot, the dermis tends to be even more sort of stapled to deeper structures. So that helps to create that crease, okay? And we can see flexure lines mostly on the soles of our feet, on our palms, on our wrists, okay? If we're getting our palm red, what we're really getting red are our flexure lines. All right. Yay! Slide 27. Even more. That's right, lines. And because PowerPoint doesn't always translate well in Notability, which is what I'm using right now, part of what you need is obscured, but of course this would not appear if you were printing out the slide. But mostly what you need on the slide is shown. If we stretch the skin, especially if we stretch it quickly, faster than it can actually provide us some give, then we can tear the underlying dermis, leaving behind scar tissue that you and I call stretch marks, but they're really called striae. Okay, very commonly formed if someone gains a lot of weight very quickly. For instance, if they get pregnant. Yay. All right, slide 28 is actually a big deal. We're going to be here for a while, and this is definitely a slide you want to focus on, okay? There are 3 pigments that are responsible for skin color. Melanin, made by melanocytes. Carotene, which we get primarily in our diet, in fruits and vegetables that are especially yellow, orange, or red. They'll typically have a lot of carotene. So like if you're eating a lot of red pepper, then you're getting a lot of carotene. If you're eating a lot of carrots, then you're getting a lot of carotene. Okay. Hemoglobin, and hemoglobin is the pigment that binds to actually heme is the pigment in hemoglobin that binds to O2. The hemoglobin molecule also binds to CO2, but not via that pigment. Okay, and hemoglobin, you probably already know, is red because the metal ion that sits at the core of the 4 subunits of hemoglobin is iron, and when iron is exposed to oxygen, it's red. Okay, melanin isn't one color. There are 2 major types of melanin. There's kind of a reddish yellow, kind of like a brick red or an orangey kind of color, and another type that's brownish black. Okay, and most people make both types. All right, but we don't all make those 2 types of melanin in equal amounts. All right, so if I were to make far more of the reddish yellow melanin variety, then my skin would be more reddish yellow. And if I were to make more of the brownish black variety of melanin, then my skin would be more brownish black. All right. Therefore, skin color reflects not just how much carotene I'm getting in my diet, not just how evident my hemoglobin is seen through my skin, okay? Seen more so through my skin if I blush. That's what blushing is, okay? But also which variety of melanin I possibly make more of. Not only that, but lysosomes, which are vesicles, okay, but they're specifically vesicles that contain within our cells digestive enzymes. We want those digestive enzymes to be present but contained because we want to be able to pick and choose when things get broken down. We don't want all of the innards of our cells broken down all the time. And lysosomes, those really the enzymes inside lysosomes, they break down melanosomes. In other words, melanosomes don't persist forever, all right? However, some people have melanosomes that persist longer. They stay around for longer. And some people have melanosomes that get digested fairly quickly, Okay, so that's another determinant of skin color. All humans have the same number of melanocytes, okay? But we don't necessarily make the same amount of melanin. So where melanin is concerned, we can consider which form, the reddish yellow or the brownish black, is more abundant. We can consider how long those melanosomes are allowed to persist before they're digested, and how much melanin is made, okay? If someone has especially dark skin, then they make more melanin, they make darker melanin, and they retain their melanin for longer, all right? So lots and lots of determinants of skin color. Hopefully you can smell a list opportunity. Let's move to slide 29. Carotene, again, is a yellowish orange pigment that we get primarily via our diet. It can be converted to vitamin A and is therefore especially important to, for instance, visual health. Maybe you have heard that before. And carotene tends to accumulate in the stratum corneum. You can see carotene best through the palms and the soles, all right? Hemoglobin, again, a reddish or pinkish hue. It's the protein that includes the pigment that binds to both oxygen and carbon dioxide. Slide 30, and for that matter, 31, we'll discuss homeostatic imbalances. Sorry, the dog's making weird sounds. Okay, so slide 30 is a concept or a set of concepts that Tessa really likes. Wink, wink, nudge, nudge. We can argue that being exposed to the sun is a good thing. And we can argue that being exposed to sunlight is a bad thing. There are two advantages to being exposed, at least somewhat, to sunlight. We hopefully can list both of them already, but I'll help. If we're exposed to sunlight, then we are ensuring by telling our keratinocytes in stratum spinosum to take up melanin and coat that sunny side of the nuclear envelope, that our DNA in those keratinocytes is protected. We're ensuring that the next exposure is less likely to cause damage via UV, all right? And then the other advantage of exposure to sunlight, we're probably especially aware of because of where we live, is to start that vitamin D synthesis cascade. Remember that exposure to sunlight converts precursor A into precursor B within the epidermis. And without that exposure, then we're not going to even start that ball rolling, which is no bueno. However, excessive sun exposure can be very bad news, as you hopefully already know, but chances are you don't know exactly why. One reason why excessive sun exposure is, bad news, is that exposure to sunlight and UV causes elastic fibers in primarily the dermis to clump together. And that pulls on the skin in the regions of those clumps, making the skin appear leathery. So there's an aesthetic issue with excessive sun exposure. Excessive Excessive sun exposure. I think I was trying to make sun and exposure one word. Can also suppress or depress, make less efficient, make less viable the immune system. Yeah, just temporarily, but still, no thank you. That's on my list of no. Okay, and excessive sun exposure means that we're exposing ourselves also to excessive UV. UV can alter DNA, in other words, cause mutations. And mutations tend to beget mutations. In other words, if DNA mutates once, it's much more likely to mutate again. And if it mutates twice, then it's much, much more likely to mutate again. And if it features 3 mutations, then it's much, much, much more likely to mutate again. So we don't even want to chance it, all right? Because if DNA becomes mutated, that increases the likelihood that those mutations will code for the regulation of the cell cycle. And in other words, the genes that code for ensuring that cells don't grow out of control. And when cells grow out of control, that's called cancer. That's what cancer is. It's out of control growth. All right. So UV damage can lead to cancer. So no, thank you. Another issue with excessive sun exposure in excess will denature folic acid. And folic acid is necessary for DNA synthesis, which is especially important in people that are growing very rapidly, like embryos. Therefore, it's especially important that a gestating woman not expose herself to excessive sun. One thing that's not on the slide, but it's pretty obvious, sunburn can be quite serious. It's not strictly a first-degree burn. That's another obvious hazard of excessive sun exposure. Also, excessive sun exposure can increase our skin's sensitivity to, for instance, products or medications that would otherwise not harm us. So, for instance, we might develop a rash in response to our laundry detergent following excessive sun exposure, whereas had we not exposed ourselves to that excess sunlight, then laundry detergent would have no significant impact on our skin, okay? And that increase in the skin's sort of irritability or sensitivity to different medications, different maybe applications is called photosensitivity. Excessive sun exposure can make us more photosensitive, in other words. So, wow, that's a fairly long list of sort of the negatives to sun exposure. Moving on to slide 31, there are many different colors that may be expressed in the skin. And if we see a certain color shining through or a certain tone shining through, then we can sometimes predict what the underlying issue might be. So, for instance, if someone presents with cyanosis, then their skin has a blue hue. That's very frightening because that means that their hemoglobin molecules are not being exposed to O2, to oxygen. They're not getting enough oxygen into their blood for whatever reason. Very, very dangerous. Erythema or redness can be an indication of fever, of hypertension, of inflammation, of an allergic reaction. Pallor, meaning paleness, and that one can be difficult to pinpoint. Usually the patient has to either present a photo of their normal skin tone or confess that they appear quite pale. And pallor can be indicative of anemia and or low blood pressure. Jaundice is a yellowish skin color and can be evident even in the whites of the eye, usually indicative of a liver issue. Bilirubin is converted, or I shouldn't say converted, heme is converted into bilirubin in the liver. And if that isn't going well, then some of that pigment can spill over into the blood and be expressed across the skin. Okay, so jaundice, liver issues, bronzing. Again, the patient may have to report that their skin has changed color. In this case, may be indicative of a lack of steroid hormones, and steroid hormones include some of the hormones made by the adrenal glands. Certainly the hormones made by the gonads, all right? Or bronzing may be indicative of a pituitary gland malfunction, which may mean that the pituitary gland is obscured by cancerous growth, okay? And then a bruise or a hematoma is caused, as you probably already know, by clotted blood that's sitting underneath the skin and hasn't been fully cleaned up yet by macrophages. So it's indicative of blood loss, at least temporary blood loss. Okay, and I believe, yeah, that's the last slide of this particular